Hello, everyone. Thank you for attending today's webinar, GeneTox, a pivotal part of safety assessment, presented by Frontage Laboratories. I'm Liz Wesky, and I'll be moderating this webinar. Our speaker today is Javed Bali, PhD, Director, DMPK, Safety and Toxicology of Frontage Laboratories. You can read his full bio on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please ensure that your volume is all the way turned up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the questions and answers tab on the left side of your screen. Okay, now let's begin. Javed, please go ahead.
Thank you very much, Liz. Welcome, everybody. Good morning, good afternoon, or good evening, whatever is your geographical location. So we are going to present today about the genetic toxicology as a pivotal part of the safety assessment. Before going and diving deep into this webinar, I would like to give a couple of definitions. The first one, what's a toxicology? Toxicology is considered as a science of poisons. It is the study of adverse effects of chemical substances on living organisms. Toxicology is a relationship between the dose and its effect on the exposed organisms. Toxicology is an interdisciplinary science which overlaps with other branches of science, especially biology, chemistry, pharmacology, and medicine. Because we are going to talk about genetic toxicology today, so we need to know what the genetic toxicology is. It's a study of chemical, physical, or biological agents that can cause damage to the DNA. The damage to DNA can be at two levels, either at nucleotide level or at chromosomal level. And this damage can be induced either by direct mechanism or the indirect mechanism. When we talk direct mechanism, that is the chemical or the metabolites which interacts directly with the DNA and cause either the change in the sequence of nucleotides or cause the change in the structure or number of chromosomes. Whereas the indirect mechanisms could be other than direct interaction with the DNA, like it can be an interaction of a chemical substance or one of its metabolite with the mitotic machinery or the cell machinery like spindle fibers or the replication enzymes, and they can also cause a similar kind of damage like DNA damage or damage to the chromosomes. This pictogram will give you a little more deeper insight of genetic toxicology and what it does and why we need to study the genetic toxicology. And on your very left side, there's a DNA molecule. And this DNA molecule has one nucleotide sequence here, which is a base pair of AT, that's adenine and thymine base pair substitute base pair. And if there is a DNA damage, like any molecule, any compound which come and hit at this particular nucleotide sequence, what can happen? There are two ways what can happen. We know that there is a large number of enzymes which operates on the DNA, and always there are DNA damages either because of our exposure to chemical substances, exposure to radiations, or sometimes there are changes during the DNA replication, and those all changes or damages get repaired. So if this is a complete repair or it is an error-free repair, mean the DNA becomes exactly the similar as it was in the beginning. So that repair will complete it and there is no damage. Or sometimes the damage is that much that it kills the cell. That's called apoptosis. So in both cases, either it is a complete repair or if it is a cell death, there is no consequences. There is no genetic toxicology. There is no genotoxicity, and the DNA either get fully repaired or the cell died. So that cell is not there to cause any damage. However, the problem occurs when there is a misrepair or when there is a no repair. For example, if there was a DNA damage at the adenine thymine nucleotides sequence, And because of a mutation, it's called point mutation, that it changed the thymine nucleotide to guanine nucleotide. So it becomes AG. And when this DNA will replicate, the one DNA strand will make an A and T DNA pair, whereas the G becomes C. So the G will binds with the C. So there is a complete change in the nucleotide at that particular point. So this is called a point mutation. So if a G get change, T get changed with G, so it become a GC base pair. So that base pair can cause different types of damages. If it is, it can be a nonsense codon nucleotide change, or it can be a missense nucleotide change, leading to different types of consequences later on. Or second option is that there's a chromosome breakage. So you can see there's a two on your right side of the screen. You can see the two fragments of the DNA chromosome. So the whole chromosome became broken into two fragments. That's a chromosome break. Then we call chromosomal aberrations. So chromosomal aberrations could be addition, deletion, inversion, or translocation. So you can see in here, there was a breakage in chromosome, and then the broken fragments get attached back to the original segment, but it did not get attached exactly the same way. It may get inverted and get attached to the original fragment. So suppose if there is a gene sequence of A, B, and C, it gets reverted and attached back to the original fragment. So instead of ABC, it become ACB. So that whole sequence of the genes get changed. Or it can be a translocation, that one fragment get broken down from one chromosome and get attached to a different chromosome within the genome. Suppose it is a broken down fragment of chromosome number 3 and get attached to chromosome number 11, for example. So that can lead to the consequences. So this is a bigger picture of what the genetic toxicology can does. And when this happens, these are the areas like point mutation. If it is a chromosomal breakage, if there's a chromosomal aberrations, they can lead to cancer. So all cancers are a result of mutations, or you can say majority of the cancers are a result of mutations, but all mutations do not lead to cancer, because some of them get repaired completely, or in some time there is apoptosis, which is cell death. So in those cases, the cell is not available to get into a cancer cell. But whenever there is a cancer, there are more chances that it is coming from mutations. So in the past, what the genetic toxicology was and how we use the GeneTox in these days is an overall risk assessment paradigm. For example, we have a hazard identification. First, we need to find that if a compound, any of the compound, if it can cause damage to the DNA. And if we say, yes, it can cause damage, then we need to find at what dose, because what's the concentration of the nucleotide, any test article at which it can cause damage to the DNA. Once we determine that, okay, there is a hazard from a compound, a particular dose of the compound can cause damage, then we need to do the risk assessment. That what's the probability of a human exposure at that dose? Do human exposed to those particular test article at that dose, or do we expose at a lower doses? And then based on our hazard identification, exposure assessment, and risk assessment, we do the risk management. We can manage our exposure to any particular substances which can cause damage, and we can reduce the damage to that. In the past, GeneTox was just was used as for binary outcomes, like if there is a DNA damage, yes or no. So there was no full risk assessment paradigm. Only we were doing like hazard identification, that if a compound is can cause gene tox, or if not. But now we use this whole data for the risk management decisions. Like we know the compound, it can cause mutation. We know a particular dose can cause mutation. Then we study, okay, what's the human exposure levels? And then we can manage to reduce the exposure levels. So this is what the GeneTox is used these days with advanced information and everything. Here is the overall drug development paradigm. We convert the drug development process into 3 different phases. It was called discovery, development, and commercialization, or we call it bench to bedside. So discovery is when we do a bench work in the laboratory, and commercialization is the bedside, when the drugs are available to patients in their bedsides. Discovery is mainly because of through the research, and development has two major phases: clinical, preclinical, and clinical. So preclinical is, that's where the genetic toxicology works. So in the preclinical area, we do the genetic toxicology, we do safety pharmacology, repeat dose general toxicology, and all other toxicology studies, which all are in animals. It can be in vitro or it can be in animals. And when we use animals, it can be rodents or it can be non-rodents. So that's the area where we fall, especially for the GeneTox. So you can see the GeneTox acts at the very early stage of the drug development. Although it is a very small portion of the drug development full paradigm, but this is a very important and pivotal part, because first we need to know if the compound is DNA reactive or not. If the compounds are DNA reactive, we can't take them into drug unless it is a cancerous drug, or until we have has a risk management or risk assessment for that. So going further into the what different areas of GeneTox, this is a list of standard assays used in genetic toxicology, and what different endpoints do they measure. The first assay is called Ames assay. It measures the point mutations, which you saw in the previous spectrogram, adenine to thymine or adenine to guanine mutations. So these point mutations, it's measured by Ames assay, in vitro and in vivo micronucleus assay. These are the two micronucleus assays which measure the structural or numerical aberrations in the chromosomes. In vitro is done in vitro through different cell lines, and in vivo is normally done in rodents, especially rat. Then we have another in vitro assay is called in vitro gene mutation assay. Mouse lymphoma is used the mouse lymphoma cell lines. This assay is unique in features because it can detect both point mutation and structural aberrations. Comet assay doesn't fall into any of these categories because it does not detect actually mutations or aberrations, but it does detect DNA strand breaks. Some of these DNA strand breaks do come from oxidation of the nucleotides or due to structural or changes, but ultimately what it detects as an endpoint is the DNA strand breaks. It could be a single strand break or it could be a double strand break. Then the PIGA assay and the Transgenic Rodent assay, these are the two assays which also detects the point mutation. We will go into more detail on all of these assays. Then out of all these, you can see only the first top three assays, Ames, in vitro, and in vivo micronucleus. These form the core battery. And I will talk about the core battery little in detail little later. But the core battery is what is the requirement, the minimum requirement for studies to conduct and submit that data to the FDA or any of the regulatory agency for the review of your data for drug submissions. There are few international regulatory guidelines which operates or which which determines what assays need to be used. The first one, or the most important one, is the ICH S2R1. ICH is the International Conference on Harmonization. S is for the safety, so it has a guidelines for the safety and revision 1. This guideline was approved in June 2012. This guideline says that there are two options to conduct the overall assays for genetic toxicology, or you can say there are two options for genetic toxicology testing. Option 1 is 2 in vitro assays and 1 in vivo assay. And when we do 2 in vitro assay, the first in vitro assay is the Ames assay, which is through bacteria. The second in vitro assay is cytogenetic assay. It could be micronucleus or MN assay, could be a chromosomal aberration assay, or could be an MLA, which is mouse lymphoma assay. These all three are conducted through different types of cell lines. Chromosomal aberration and mouse lymphoma used to be the assay of preference in the past, but currently the micronucleus assay is the most favorite or most preferred assay. And then we do the third assay, which is in vivo. So the 2 in vitro are Ames and cytogenetic or micronucleus, and the 1 in vivo is the in vivo micronucleus. The option 2 of the ICHS2R1 guideline is doing 1 in vitro and 2 in vivo assays. So but again, when you do the option 2, the first in vitro is again the Ames assay. So you can see the importance of this Ames assay. Whether we choose option 1 or option 2, you have to conduct the bacterial reverse mutation assay or Ames assay. And in option 2, the second it says 1 in vitro and 2 in vivo assays. So the first in vivo assay is the in vivo micronucleus, again the same, which is the part of option 1. And then we do the second in vivo assay. But there is a caveat that the second in vivo assay should be in a different tissue. In vivo micronucleus assay, when we can conduct it either through bone marrow or through blood. But whatever we use, either bone marrow or blood is also a product of the bone marrow. So it means it is the same tissue. So when we conduct the second in vivo assay, it should be other than blood or bone marrow. So currently there is a one assay which can be conducted in standard rodents, or you can say the non-transgenic rodents, and that assay is called Comet assay. We can conduct Comet assay in any of the tissue, and I will talk about the detail about the Comet assay in a little later. So there are two options. Option 1, 2 in vitro, 1 in vivo assays. Option 2, 1 in vivo, 2 in vitro assays. We'll go into detail of each of these assays now. The Ames assay, which is the first one, this assay was developed in 1970s by scientist Bruce Ames, and that's why it's called Ames assay after the name of the scientist. This Ames assay is still act as a gold standard for genetic toxicology. It was developed like more than 50 years ago, but still is a gold standard. As in the previous slide you saw, if we do either option 1 or option 2, you have to conduct the Ames assay. So this is one of the basic requirement. This assay has its own regulatory test guidelines, how to conduct the assay. These regulatory test guidelines come from OECD, which is Organization of Economic Cooperation and Development. It has a test guideline for each of these assays, how to conduct the assay. The guideline for Ames is OECD 471. There are 5 different strains of bacteria which are used to conduct the assay. Like as a standard, you can use, and I am naming these 5 strains here, but there are some other options which we can use. But these are the ones which are standard according to the OECD 471 test guideline. Out of these 5 tester strains, 4 are Salmonella typhimurium strains. They are TA98, TA100, TA1535, and TA1537. The second or the 5th strain we use, that is from E. coli, Escherichia coli, and the strain is WP2 UVRA. So TA98, TA100, and why we call them the TA and this one. So the TA name came from a word Typhimurium Ames. This is the assay which I first time used in my undergrad when I worked on the leather tanning industry affluents, because those industries, they use chromium, and chromium is one of the mutagenic. So I worked on that. And since then I was looking why we call them TA. So that was one of my curiosity. I tried to keep finding, but I never found what the TA stands for. Then I met Dr. Bruce Ames at one of the SOT in 2011. And I followed up with him and I asked why we call this the TA. So he was amazed. He said, "Oh, nobody ever asked me this question," but then he explained me why it is called TA. So that was a long story. So he told me that he said initially his lab folks, they named these strains as BA98, BA100, and like that. But BA stands for Bruce Ames. But he said, I thought it is too personal with my name. So he said, I suggested to name them as TA, coming from Typhimurium Ames. So this is one of the history behind TA. I think very few people know that. So these strains, they all have a pre-existing mutations. So they are mutant strains, and when we treat them with the test article, they get another mutation called. So that's why it's called reverse mutation assay, because we reverse the mutations. Salmonella typhimurium strains, these 4 strains, they have a mutation in their histidine locus, whereas E. coli strain has a mutation in their tryptophan locus. So those bacteria which are mutated, which cannot synthesize their all essential nutrients, they are called as auxotrophs. these auxotrophs, we provide them as traces of histidine or tryptophan. They are medium, so they can grow for a few divisions. And if they get mutated during those divisions, they will keep growing. Um, so the TA98 and once they keep growing, they will form colonies, and that's from auxotrophs, they will become prototrophs, and that's how we detect them. As we talked in the previous one of the slide, that the AIMS assay, it detects for the point mutations. We need to find why we use 5 strains. So the TF98 and TF1537, these are the two strains. They find exactly the same type of mutations, and this is frame shift mutation. And the other 3 strains, they detect the base substitution mutations. So that's why we use 5 strains. But out of these 5, TA98 and TA100, only these 2 strains, they can detect 93% of the mutagens of any chemical substances which can cause mutation. These just 2 strains can detect 93% of the mutation causing compounds. So, but just to find it 100% accuracy, we use some additional strains. So that's why TA98 and TA100, if you are doing only screening of your studies, you can just use TA98 and TA100, and you will get to like 93% correct answer. According to the OECD 471 test guideline, the highest concentration of any test compound we need to test during the AIMS assay is the 5,000 microgram per plate. And then we use different other lower concentrations, so about 6 to 7 different concentrations between 5 to 5,000 microgram per plates are used in the laboratory, and see what kind of mutations, if they can cause any, we can detect. The criteria for the positive outcome for TA98, TA100, and WP2 UVRA should produce more than twofold or higher, greater than twofold number of revertant colonies in the plates compared to their concurrent control. Whereas for TA1535 and 1537, the criteria is greater than threefold. And the reason between the difference of these twofold versus threefold is because the TA98, TA100, and WP2, their spontaneous revertant colony count is a little higher. 98 and WP2, they normally produce between 20 to 40 revertants per plate, whereas TA100 is normally more than 100 revertants per plate. Whereas for 1535 and 37, the standard number of revertants is normally between 10 and 20. Sometime, most of the time, it's below 10. So because when you see a very small number of revertant frequency, so that's why the fold criteria which was established with the OECD test guideline is threefold for these 2 tester strains. This is like a pictogram of overall how we conduct the assay. We have the plates with which have agar, and they are minimal agar plates. They do not have, for salmonella strains, they do not have histidine, and for E. coli, they do not have tryptophan in them. But we just add a few traces of these histidine or tryptophan, so the bacteria can grow for a couple of divisions. Then we have another tube called top agar. It gets, it is used for in the melted form or molten form. We add the test article in this one. We add the tester strain in this one. Then we can add S9 with and without S9. So just to have a metabolism outside the living off system. These all get mixed, and then they are poured on the plates. So all bacteria will start growing because we added histidine into the plate. But after a couple of divisions, the histidine will exhaust from the plate, so the bacteria will stop growing. However, those which get mutated during that time will keep growing. And once they keep growing, this is how the colonies will form. So once we plate the top agar on these plates are incubated at 37 degrees Celsius for about 52 to 60 hours. And then plates are counted using the automatic colony counter. So these are the 2 plates. This one was treated with the negative control, and this one was treated with the known mutagen. So you can see these dots in these plates on your right side of the screen. And the plate at the top has a much higher rounded colonies compared to the plate at the bottom, which is a control one. So the control one has a revertant normal colonies, and top one has a induced revertant colonies. So that's how we differentiate between uh, mutagenic or non-mutagenic compounds. So this is how the Ames assay works. Then going to the second, which is again the part of the core battery, is the in vitro micronucleus. Again, this assay has its own OECD test guideline, which is OECD 487. Anybody who wants to study more about these guidelines, you can get these guidelines from the OECD website. This assay, the micronucleus, it investigates for the chromosome breakage or spindle fibers. In the initial one of the slide, I mentioned that the DNA or chromosome damage can be direct or indirect. The direct damage is the clastogen, which acts, clastogenic are the compounds which acts on the chromosome and cause breakdown of chromosome fragments. Whereas angiogenic compounds are the chromosome which do not act directly on the chromosome, but they do act on the spindle fibers and during mitosis. And once the spindle fiber is broken, that chromosome cannot move towards the opposite poles or become part of the daughter nuclei. So this is how those micronuclei are formed in the cell. The top dose for this type of assay is 500 microgram per ml or 1 millimolar, whichever is the lower. So the millimolar is determined based on the molecular weight of the compound. If the compound molecular weight is suppose 930 or anywhere above 500, then we will have a top dose of 500 microgram per ml. But if the molecular weight of a compound is 350, then the top dose would be 350 microgram per ml. Test is conducted in different test conditions. The two test conditions are normally use either 3 hour or 4 hours. Some labs they use a 3 hour treatment. Some labs use for 4 hour treatment. In the presence and absence of S9. S9 again is a metabolic activation which converts a test article or any compound into its metabolites. The reason for that, why we use it, because sometimes the original compound is not mutagenic or genotoxic, but one of its metabolite is. So we convert it into its metabolites and see if it can cause genotoxicity after getting metabolized. So they are treated with and without S9 in the short-term treatment, suppose after for 4 hours. And then after 4 hours, the culture media are washed to remove the test material from the culture. And then a fresh medium is added and cells are put back in the incubator for another about 20 hours. So the overall incubation period is 24 hours. In the third test condition is 24 hour without S9. So because if we keep the S9 for a longer time in the cells, it is toxic for the culture, for the cell culture or for the cells. So it should be washed out after 3 to 4 hours. But if we do the 24-hour treatment with the test article, this is without S9. Cells, they stay in the medium along with the test article for the whole duration. And then at the end, cells are washed, material is removed, test material is removed, and the cells are then counted or analyzed, howsoever we are doing that. The top concentration we use for testing, as I said earlier, is 500 microgram per ml or 1 millimolar. But when we evaluate for the chromosomal damages or for the presence or absence of micronuclei, the top dose we evaluate is 50 at the 50% or approximately 50% cytotoxicity. And what that mean is cytotoxicity, because when we treat the test article with the cells, the test article can inhibit the cell growth based on its cytotoxic nature. So if we evaluate any cells at higher than 50% cytotoxicity for their chromosome damage, that chromosome damage may come from the cytotoxicity and not from the genotoxic nature of this compound. So that's why, according to the guidelines, we only evaluate the cells or the cultures which have produced approximately 50% cytotoxicity. There are different types of cell lines which are used for that. HPBL is a human peripheral blood lymphocyte. We directly take human blood. We trigger the blood with the phytohemagglutinin, which is a compound which triggers white blood cells start growing. Normally, white blood cells which are in our peripheral blood, they are dormant. They are not multiplying. but we trigger them and they start multiplying, and that's what we use for that assay. Or the TK6 cell line, Chinese hamster ovary CHO cell line, or L5178. This is the mouse lymphoma cell line. So these different cell lines are used. In vitro micronucleus assay can be performed either through the microscopic method, which is a conventional method, and or by the flow cytometric method, which is the most advanced method, and most of the labs, they are now moving towards the flow cytometric method. We at Frontage, we conduct this assay by using flow cytometric method. There are a few advantages of doing the flow cytometric analysis. The number one is the fast turnaround time, because we don't have to prepare slides, we don't have to score slides. So if we are treating cells in like one day, we can have a data by the end of the day next day. Instead, in the microscopic method, normally duplicate cultures are used. For the flow cytometric method, we can use triplicate cultures for each test concentration, and then we score 5,000 nuclei to detect the frequency of micronuclei, compared to 500 nuclei which are used for in the conventional method. So this is more robust, statistically robust, and a fast turnaround time. And so that's one of the few of the advantages of using the flow cytometric analysis. This is again a small pictogram how the cells look like, or what's the phenomenon of the micronucleus assay. We have a parent cell. It is undergoing division, and during the division, if this cell come in contact with any of the compound which can cause genetic damage, you see there is an acentric fragment. So this chromosome gets broken down, and this small fragments get separated from a chromosome. Now at the end of the mitosis, there's a around telophase, the two nuclei are formed within the main nucleus. So it's a binucleated cell, and it has a small fragment here, you can see, and this is a broken down fragment of chromosome. This is called micronucleus. Especially for this type of assay, and when we are doing a microscopic method, at this stage we add a chemical called cytochalasin in the medium. Cytochalasin is considered as a spindle poison. It lets the cell grow through karyokinesis but stop the cytokinesis. Cytokinesis is the process when a binucleated cell divides into two daughter cells, and each daughter cell will have one nucleus. And these are the real images from this binucleated cell. So you can see there's two binucleated cells, and the top binucleated cell has one small dot, which is a micronucleus, whereas the lower binucleated cells have multiple 3 or 4 dots here. So these are the micronuclei. So this is what we count. We count the number of binucleated cells, and then we count how many binucleated cells have a micronuclei, and that frequency is used to get the outcome of the assay. In the flow cytometric method, which we have here, you can see on the left side, this panel is for the vehicle control, and the right side panel is for the positive control. We use different gating strategies to separate the micronuclei and the total number of nuclei. In this flow cytometric method, we do not score for the cells, but we lyse the cells and liberate the nuclei, and then we count for the nuclei, and then we count for the micronuclei. So we see how many micronuclei are per nuclei, because we know there is one nucleus per cell. So as many nuclei we count, it means we are counting as many cells. And then we use the beads, which are used to determine the cytotoxicity of that. So if you look at the for example, if you're looking on the left side of the panel, and the top right corner of this left panel, there is beads. So the yellow dots, you can see these are the number of beads we have that. So that's how it is going. And then when we are looking for the micronuclei, you can see here in the circle, the top square has the nuclei and the bottom square has a micronuclei. So we are scoring, as I said in the previous slide, we score 5,000 nuclei, and then here you can see how many micronuclei are there. This is for the vehicle control, but if you look on the positive control in the same cytogram, you can see a lot more micronuclei in this micronucleus gate. So these are what we have here. We call it gates or gating strategies. So this is how we make the gating strategy to count for the nuclei and macronuclei. So this is one of the way how we can process the micronucleus assay. So looking on these, we count the number of nuclei, and then we calculate for the percent of nuclei. So that percent nuclei will give us the total overall outcome. We can run the statistical significance on this test compared to the vehicle control. If the result is statistically significant, it is considered the compound is positive. It should also produce a dose response. Then your data should be is compared with the historical ranges and things like that. So that's what the in vitro macronucleus is. When we go to the in vivo micronucleus, that's also kind of the same thing. The only major difference is it is done in living systems, like in rats. So this assay has its own separate guideline, is OECD 474. This test guideline explains how to conduct the assay. Again, it detects the same thing, macronucleus in the living system, and we know how those macronuclei are formed, because they are formed either by this breakdown of chromosomes or a breakdown of spindle fibers. This test can be performed either through bone marrow or through blood. If we are conducting this through bone marrow, animals are treated for 2 days, day 1 and day 2, and then on day 3 they are euthanized and the bone marrow is collected. If the assay is being performed through peripheral blood, animals are treated for 3 days and the blood is collected for day 4. And the reason for that is because what we are going to look into this assay is the polychromatic erythrocytes or reticulocytes. And these are the younger population of RBCs. They are formed in bone marrow. So that's why if we do the bone marrow assay, we can harvest the bone marrow on day 3. But if we're gonna do it through blood, blood has to be formed in bone marrow or come in the peripheral blood. So it takes a little more time to come in there. The highest dose for this assay is a 2000 milligram per kilogram per day, or the maximum tolerated dose. So if we do not have any information about the maximum tolerated dose, then it is always suggested that the laboratory should run an MTD study or maximum tolerated dose study before actually treating the animal for the micronucleus evaluation. So the top dose is 2000, and then we have two lower doses. One is 1/2 and other is 1/4 of the top dose. This assay can easily be also integrated into repeat dose Genotox studies, for example, 28-day studies. And if we are integrating into a repeat dose study, animals are treated as normal as for a tox study, and blood or bone marrow is collected on day 29. So if we are treating animals only for 2 or 3 days, then the top dose is 2000. But if we have to dose animals for a longer duration, longer than 14 days, then the top dose should be 1000 milligram per kilogram per day, or the maximum tolerated dose, whichever is the higher, whichever is higher. Or not higher, in case like 1000 milligram is the limit dose. But if a maximum tolerated dose is like 200 milligram per kilogram, we don't have to go to the 1000. Again, so when we get there to, if the compound is positive, then what to do? So are those micronuclei being formed as a result of clastogenicity or angiogenicity? So those are called as a mechanistic study. So we can run the mechanistic study, which is a process called FISH or CREST. So they are conducted, and I'll talk about that in the next slides. Again, this assay can be conducted through microscopic or flow cytometric, and the same as for the in vitro. So this is one of the stem cell in the bone marrow, and this cell was dividing when it got hit with a compound, and that compound caused a breakdown of chromosome fragment. So you can see the two nuclei are formed, and then there is a smaller fragment of broken down chromosome. When we did the in vitro micronucleus, we stopped cytokinesis. So we let the cell stop at this stage, and we cease the further cell growth. But because it's going in the living system in the rat, so we can't do that in this case. So we let the cell divide into two daughter cells. So the two daughter cell has both both cells have a single nucleus, but one of the micronucleus went into one cell. So it is on the top of this one. You can see there are two cells, or one with the both with one nucleus, and one has a small dot is a micronucleus. Then mm, the red blood cells, they do not have a nuclei. So this we are performing this assay through red blood cells. So initially these cells have a nuclei, and then nuclei are exuded out from the cells. So you can see the extrusion of these nuclei. And finally they become a polychromatic erythrocyte. So they do not have a nucleus, but they still have some of the RNA in there because they are freshly formed red blood cells, and they also have some micronucleus in that. So if you look in the last panel, where are there four cells, the top cell has a one dot, which is a micronucleus, and the lower cell doesn't have a dot, so there is no micronucleus in that. So this is how we differentiate the polychromatic erythrocytes with micronucleus, or polychromatic erythrocytes without micronucleus. On the lower panel, if you see, and that is the result of angiogenic effect. The top one is a clastogenic, and the lower one is the angiogenic effect. In this one, it's exactly the same process, but in again the micronucleus, you will see a small whitish dot, and that whitish dot represents the centromere. So when it represents a centromere, it means this is the whole chromosome. So, and from the previous slide, but now you have understood how these are formed. So it means these micronuclei are formed as a result of the breakdown of spindle fibers, mean they are the whole chromosomes and not just the fragment of chromosomes. So this is how we can differentiate based on the microscopic method. If we go to the flow cytometric method, as I said in the previous slide, the polychromatic erythrocytes, they're also called as reticulocytes. So we target reticulocytes, and reticulocytes have one specific antigen protein attached to their cell surface called CD71. So we target CD71 protein to see if there is the presence or absence of CD71. If they have a CD71, it means this is a younger population of reticulocytes. And if they have a micronuclei, we also label these cells with the propydium iodide, which is a nucleic acid strain. If they have a chromosome fragment, which is a DNA actually in there, so they will fluoresce at a higher channel on the propydium iodide. So that's what you can see here. If we're looking on the vehicle control panel, you see in this circle there are very few dots. So these are very few micronucleated reticulocytes. But on the other side, if you look on the positive control, you see a lot more dots. So looking on these presence or absence of micronuclei, there are like 5 animals in each dose group. We take the mean of 5 animals, and then they're compared with the vehicle controls. And based on the statistical significance, it is considered if there's positive or negative. So these are the 3 assays we talk about in the core battery in the beginning, when we talk in the beginning that there are 3 out of a long list of GeneTox assays. There are 3 which are the core battery. So these 3 forms the core battery. Mouse lymphoma, which I said was used to be one of the assay of preference in the past, is rarely used now, but sometime, for example, if we are looking for an antibiotic, which are very toxic to bacteria, so running a bacterial mutation assay with an antibiotic is a challenge. But the OECD 471 and ICHS2R1 test guidelines, they both talk still, even if you can run the, if you're testing an antibiotic, you cannot test it at higher concentration because it will kill the bacteria, but you still have to do at lower concentration because sometimes some compounds are genotoxic at lower concentrations. But if you are running an antibiotic, the mouse lymphoma assay is another assay of choice for that. This assay is conducted with a cell type called L5178Y. This cell line is a heterozygous for a thymidine kinase gene. And this assay has its own OECD guideline, which is 490. This thymidine kinase gene synthesizes an enzyme called thymidine kinase enzyme. And this enzyme is responsible when DNA replicates in the growing polynucleotide chain. It adds a thymidine nucleotide to the growing chain. If the cells are heterozygous, when we grow these cells, we let them grow for a while, and then we add a thymidine analog. It's called trifluorothymidine. or TFT. TFT is a very toxic compound. So we add TFT into the culture medium. If the cell is normal, because it is heterozygous, if it is not heterozygous is actually normal. So when it is normal, it will add TFT into the growing polynucleotide chain. And once TFT is added to the chain, it will kill the cell, or it will kill the nucleotide growing polynucleotide chain. So the cell will not grow because the cell could not replicate. However, if because of the mutation, the thymidine TK heterozygous sequence get converted into TK homozygous recessive sequence, like the TK gene become inactive. And once it is inactive, it will not be able to add the TFT into the cell, and the cell will survive in that case. So this is how it looks like. If it is a TK plus, if you look on the left side of the of this slide, if it's a TK plus and we have a TFT in this medium, the TK cell being a proficient for thymidine kinase gene, it will add the TFT and it will kill the cell growth. So cell will not grow. So we will not find any colonies on the cell. However, if you come to the middle panel, if there is a small or you can say point mutations, so it will add TFT but only to the fewer cells. And being a TK thymidine kinase negative, it will not actually incorporate the TFT. So cells will keep growing and they will grow to normal size colonies. However, if you come to the right side of the panel, and that is a bigger damage, so that it can be a chromosome damage. The middle one is a point mutation. The right one is the chromosomal damage, which is we are losing a fragment of chromosome. It means we may be losing several of the genes. And because it is a TK negative, it will not incorporate TFT. So cells will keep growing, but they will grow to a smaller size colonies. So just depending on the size of the colonies, we can differentiate if it is a result of a point mutation or if it is a result of a structural chromosomal aberration. So this is like just running one assay, we can detect both endpoints, point mutation and chromosome damage. So that's what the mouse lymphoma assay looks. So now this is the core battery. I ended with the core battery. I will just give some flair of the follow-up assays. What if you get some positive results in some of the primary or the core battery assays? The first one is the Comet assay. And we talked earlier that if we use option 2, we do the Ames assay, and then we also do the in vivo micronucleus and Comet assay. Comet assay is also called a single cell gel electrophoresis assay. And why it's called Comet assay? Because the cells, they form a comet-like shape. So that's why it's called Comet assay. The principle of this is pretty standard with the gel electrophoresis, that we create a single cell suspension from any tissue of the animals. For example, we are working on a rat and we are going to do the Comet assay through liver. So we'll take the liver, we convert that into single cell suspension, we embed those single cells into the agarose gel on slides, and we treat them with alkaline solution, so at a very high pH, about 12 to 14 pH. And then we do the gel electrophoresis. So the alkaline treatment will cause the unwinding of this in the DNA. And then when we will do the electrophoresis, the broken down fragments will start moving out of the cell. More the damage is, the smaller are the fragments, and smaller are the fragments, the longer they will travel out of the DNA head. So that's the principle of the assay. It can detect the single and double strand breaks. It can also detect oxidative damage or the direct DNA damage, and it can also detect the alkali labile sites because we use it in an alkaline medium. There are certain strengths of this assay. The first one is it can be performed in any tissue. So when I talked about the option 2, it has one in vitro and 2 in vivo assays. The first in vivo was a micronucleus assay, which is on bone marrow. So and I told there that in the second in vivo has to be in a different tissue, so other than bone marrow. Comet assay can be performed in any tissue. You can do in brain, you can do in blood and bone marrow, liver, GI tract, lung, nasal terminals. I mean, any area which you want to study. It has its own guidelines as 489 OECD. Can be conducted in vitro and in vivo, but in vitro is not a GLP standard assay. So only for the GLP studies, for IND enabling studies, you need to run only in vivo assays. It can be easily integrated into other Genotox studies. So this is a 3R friendly. 3R is a reduce, refine, and replace, where you need to decrease the use of a number of animals. So this is one of them. Image analysis system, it makes it more faster. In the past, it used to be that you stain your slides either with the ethidium bromide, or you stain with them with the GIMSA, and then you score them under the microscope using ocular micrometers. But now there are certain image analysis systems are available. You just load the slides and you select the cells to score, and it will score the cells. And then we detect for the Comet length and the percent of DNA in the tail, and they are used as the endpoints to detect the damage. And this is how you see the comets when you put them on the slide. This is how the cells look like, and so you can see that they are giving you a shape like a comet. So that's why it's called the Comet assay. And this is what we score for that. So that's the total cell. This is the head of the cell, which we initially embedded in the gel electrophoresis on the slides, in the gel on the slides, and then we did electrophoresis. So electrophoresis caused the movement of the DNA out of the head. So this was the head, and then this is the tail part. And this is calculated through the software and the image analysis system. So it calculates the size of the head, it calculates the size of the tail and amount of tail, amount of the DNA in the tail, and then the tail moment is the ratio of the DNA present in the head versus the DNA present in the tail. So these parameters tell us what's the damage, and then this is compared with the concurrent control animals. The second in vivo or follow-up assay is the Transgenic Rodent assay. And this assay is conducted in transgenic animals. It can be mutamouse, which has a LAC-Z transgene, and or the big blue, which has a LAC-I transgene. This assay has 488 OECD 488 test guideline for that. Again, it can be conducted in any tissue. Whatever tissue we can collect, whatever tissue has a DNA, it can be conducted in those tissues. But the problem with this one is we cannot conduct them in the standard or regular animals. So this cannot be integrated into repeat dose studies. This is very expensive because we use transgenic animals, and then we have several months or several weeks to get the data. So that's a little, which is like some of the weaknesses, you can say, or the of the assay, challenges for the assay. And then comes the PIG-A, is another in vivo gene mutation assay, and I would like a little more time to talk about this one because I have the privilege to be among the pioneers who developed this assay. I worked on this assay during my postdoc when I was at NCTR in collaboration with the Frontage Laboratories. So the PIG-A assay, and just let me say that it is a PIG-A, but has nothing to do with pigs. Just the name PIG is an acronym for phosphatidyl inositol-glycan. This gene is X-linked gene and is responsible for the synthesis of GPI anchors on the cell surface. And those GPI anchors attach CD proteins, CD59, 24, and other proteins to the cell surface. About more than 100 million cells are scored. Assays conducted through blood and through the red blood cells. 100 million cells are scored in 2 to 3 minutes. So it is very high throughput, very fast, robust, and turnaround time is pretty quick. So if we get the samples in the morning, we will have data in the afternoon. The good thing about this assay is that just in June, we got the OECD test guidelines approved for the assays. The OECD test guideline number is OECD 470. And the biology of the assay is that this is a sequence of genes, how they are located for the synthesis of GPI anchors. And the pig A is at the very first step of this biosynthesis. So there is a pig A gene which is involved here. And if this gene get mutated, the whole process will block and there'll be no GPI anchor synthesis. And this is how the cell would look like. On the left side is a normal cell, which has proteins attached to the GPI anchor, CD59 or CD24. And there are some other proteins which are embedded into the cell called transmembrane. If there is a mutation, there is no GPI anchor synthesis and no protein attached. The cells are labeled with CD59 and CD59 antibody, which is a fluorescently labeled antibody, and then these cells get magnetized by the use of magnetic beads, which works against the fluorescence of the CD59 antibodies. And then these magnetized cells are passed to the magnetic columns. So majority of the normal cells which have fluorescence, they get attached with the magnetic field, and only the mutant cell which pass down, and then the counting beads are added, and then the cells are analyzed on the flow cytometer, and this is how they look like at the flow cytometer. The top one are before passing them through the column, and the bottom cytograms are after passing them through the column. So majority of the cells are removed, the wild type cells, and they get enriched for the mutant cells. So in these columns, you see these are the mutant windows, which is under these circles. And the lower one on the right side is the treated column. So you can see a lot more mutant cells in the mutant window of the treated animals. Whereas if you look on the untreated post column window, you will see there are very few cells. So just looking at this, you can see differentiate between the amount of mutants. So this is how we determine the presence or absence of mutant cells in PGA assay. So the overall summary for the Ames assay: we run the Ames assay. If it is positive, if it is negative, you're good. But if it is positive, then what we can follow up that? We can follow up in the in vivo gene mutation assay, doing an in vivo gene mutation assay that can be a PGA or transgenic rodent assay. And then we confirm for the absence of mutagen. Then we also, sometimes the test article has some impurities in the API. So when we look for the impurities, then you either can purify your API. API is the active pharmaceutical ingredient. So you can purify your API to remove the mutagenic impurities and retest that. Or if you cannot remove the impurities, then you have to qualify that your impurities should be less than certain concentration in the assay. But if you can run it, if you cannot remove the impurities, then you can follow up with the PIG-A. PIG-A is one of the best options for this one. Then in vivo, if you get positive results in the in vitro micronucleus assay, there are two options. Either you do a fish mechanistic study, that is fish analysis. Fish has nothing to do with fish. Again, it's the fluorescent in situ hybridization. So we look for the presence or absence of centromere, mean if it is a full chromosome or fragment of chromosome. So, or if we cannot do that, we can follow up with the Comet assay. And if you get in the in vivo micronucleus, if that is positive, again, you follow up with the fish or crest. If it is positive for the presence of centromere, mean if it is angiogenic, then angiogens have some threshold so that we can keep it below that. Or if it is negative in the in vivo micronucleus, that is good, but we need to make sure that cells have enough exposure to the, animals have enough exposure to the bone marrow. Just to give you that there are some screening assays. You can do the in silico QSARs, which is direct and lead scope. That can be done in a very early stage in the drug development process. We can do mini or micro Ames using only 2 strains in 6 or 12 well plates. In vitro micronucleus can be conducted in screening assay only either doing 4-hour treatment with S9 and 24-hour without S9. And the multi-flow assay is another. Frontage Laboratories, they developed this assay, which in one assay you can detect multiple endpoints and it can be conducted in 96 well plates. So these are one of the screening assays we can do that. So that's all. Thank you very much. And Liz, it's back to you now.
Great. Thank you so much for that presentation. All right. So we are going to be moving on to the Q&A portion of today's webinar. We don't have a lot of time left on the clock, but that said, please do still feel free to submit your questions using that Q&A tab to the left side of your screen, because if we're not able to address your question during the live portion of the Q&A, we can follow up with you or do our best to follow up with you personally after the webinar. That said, let's dive into a couple of quick questions here. So first question for you. Have you ever found a positive hit with in vitro MN post 3 to 4 hours? Are there cases where genotoxicity occurs following a longer incubation period?
Yes, so it all depends on sometime you do get positive in the shorter term test, and the reason is because the shorter term test we can test at a higher concentration, and you don't see toxicity. Suppose if I'm treating for the 4 hour, I can see 50% toxicity maybe at 150 microgram per ml, and it is positive. But in 24-hour, 150 microgram per ml is like 70% toxic. So that because as I said in my presentation that we shouldn't be testing above 50% cytotoxicity. So the 150 microgram which was positive in the 4 hour become more cytotoxic in the 24-hour treatment. So we should not evaluate that one. So and maybe the 50% toxicity in 24-hour is at 75 microgram per ml, and 75 microgram per ml is not genotoxic. So there could be an option, or it could be a chance that you see positive result at the 4 hour, but not in the 24-hour, and that is because of the difference in toxicity at different concentrations.
Great. Thank you so much for that response. All right. It looks like we have time for just one more question today. But as I mentioned, we had a lot of great questions come in and we didn't have time to get to them all. So we will try our best to get back to you after the webinar. But diving
Sure.
into our last question for the live Q&A portion today. So how predictive are the genotox studies for human safety?
Oh, yep. So they are very predictive. As I said, the Ames assay is the gold standard. I mean, even if we see the positive result in the bacterial mutation assay, it is still considered a very predictive for the human safety, because if I'm developing a drug for inflammatory disease or for any disease other than cancer, and if the Ames assay is positive, uh, I cannot take the compound into drug development until we do a follow-up assay with another in vivo gene mutation assay. So
Great.
it means the prediction from the in vitro assay is highly prevalent for the human safety. Just in short.
Okay. Great. Thank you so much for that response. And it looks like we actually do have time for one more quick question here. So our last question for you will actually be, so could you comment on the inclusion of pig A and or other micronucleus endpoints on to repeat dose toxicology studies?
Yes, sure. So I'm a big fan of 3Rs. I am a big advocate for the 3R processes. So I would always prefer that we can easily, as I mentioned, we can easily tag on the PIGA or the in vivo micronucleus, or even the Comet assay to the repeat dose tox studies. So if we have a prediction, and normally because the gene tox studies are done much earlier in the preclinical drug development part, so if you have an in vitro micronucleus positive, or if you have an Ames positive and you want to follow up with the pig A or in vivo Comet assay, I would suggest adding on those to your repeat dose tox studies, but always make sure that you are meeting the top dose criteria in the repeat dose studies, because for a 28-day study, our high dose may not be that high, which is required for by the guideline in GeneTox. So if you are meeting the top dose criteria properly for your GeneTox, I would always suggest to add on those to the repeat dose tox studies.
Great. Thank you so much for that response. All right. Well, I would like to thank everyone in attendance today for attending this Fierce Biotech webinar and for submitting so many great questions. I'd like to thank our speaker, Javed, for participating, and Frontage Laboratories for presenting today's webinar. As I mentioned at the beginning of the webinar, this session has been recorded, and you will be able to access that recording within 24 hours using the same audience link that was sent to you earlier. Thank you again so much for joining, and we look forward to seeing you at future events.
Thank you very much.